club is a common scientific practice where scientists gather to read and discuss research articles from their field. And often one person will really deeply analyze the paper. Everyone reads it, but one person kind of takes the lead in deeply analyzing the paper and then presenting it for discussion to everyone in the group. So we use this journal club assignment in class as an opportunity for students to get really familiar with a research article, to have the experience of discussing science with their classmates, and also to practice their presentation skills. As the instructor, I have a coaching meeting with them in advance of them giving this journal club presentation to help them prepare and make sure that they're ready to present the paper. But what we noticed is that even with this coaching meeting, students were still kind of struggling with the article. When they got into analyzing the results of the article or kind of understanding the whole narrative flow of these papers, that's where they were sort of struggling. They didn't have the skills yet to do that. And also I found in this coaching meeting before the presentation, often the way this meeting went was that I would be answering a ton of questions for them. What I wanted in this meeting was more for them to be able to explain the paper to me, and then I could answer just a few questions, but then we could spend most of the time talking about the most effective way to present this paper and its narrative to their classmates in the presentation. My innovation, which I implemented with my colleague, Dr. Jordan Mobert Parker, it starts with an existing learning tool called Create. Create is a method published by Hoskins et al. In 2007, which is a scaffold process to help students read and understand scientific articles. And Create is an acronym that stands for several steps, each of which is a tool that students can use to help them dive deeper into the research. Our innovation was to use this existing learning tool, Create, but specifically as a preparatory step for the journal club process. The way that our innovation works is that students select, with instructor help, an article to read for their journal club, and then they go and do the Create process on that paper by themselves. During that coaching meeting, I would ask them to explain the paper to me using the materials they generated through the Create process. And then we would discuss the paper together, and then after the coaching meeting, they will finish prepping their presentation and then deliver the presentation to the class. And I, as you know, haven't read the paper. I skimmed it really quick when we looked at it, but I'm gonna let you all tell me about it, because after doing Creates, you're definitely the experts on this paper. Why don't we jump into the figure? So what was the first figure? And if somebody could kind of talk me through the figure, starting with the aim or the question or the goal for that figure overall.
So I did figure one sides, and figure one was
that coaching meeting. I'll often ask them to return to some of their Create materials to help explain certain parts of it, or if there's some confusion, we can look back at the analysis that they've already completed to try to clear some things up. And just so I understand the details of the methods here. So in A, this protein GP105 is green. Okay. DAPI is DNA, that's blue.
And then the cell membrane is red.
As we go through, I often am also trying to gauge their understanding to see if they've really got it, or if I maybe need to pry a little bit or dig a little deeper with them to ensure that they actually understand it. If you were the authors of this paper and you wanted to publish this paper in my journal, how would you phrase what's the big deal here? What's their big finding?
Well, the big finding is that there is a nucleotide structure, which is of course
during the students' journal club presentations, their classmates are their audience, and I ask the other students to complete peer evaluation forms of both the presentation and the presenters for a few different reasons. So A, I give them questions on this form to guide their thinking and analysis of the presentation, and also to give them kind of a structure to give constructive feedback within. And I want them to watch these presentations and reflect on what they would like to do for their own presentations, or what they think makes for a good presentation. Since implementing this Create step as part of the journal club presentations, I've definitely noticed some impacts. Previously, we would spend the coaching meeting, me almost explaining the paper to them asking me so many questions. Now that they've completed the Create process before that coaching meeting, they come and explain the paper to me, and we can have a much more productive meeting that focuses on preparing them for the presentation itself. At every coaching meeting, I always start off by asking the students, how was the Create process. How do you feel about the paper? And they generally feel it is a lot of work, and I try to be transparent with them about that. But they also tell me pretty consistently that they are glad that they did it because they feel a lot more confident now about the paper and about presenting it.